data, real-world evidence, and clinical trials. And I'm delighted to introduce the panel that we have present. Today we have Ida Sim, who is one of the Vivli co-founders, as well as a professor at UCSF. We have Marcia Levenstein, who is also a senior advisor at Vivli. We have Gregory Pappas from the Food and Drug Administration, as well as Jack Markadian from Rutgers University. I think we have a really good panel who will be full of different viewpoints, and really hope that you will take this opportunity to ask them questions as we move forward. So at this point, I'm now going to turn over to Ida Sim. Ida, over to you to tell us more about the webinar and kick us off. Thank you so much.
Thank you. Can you hear me okay?
Yes, perfectly. Thanks.
Wonderful. Thank you. Thank you so much, Julie, and thank you everyone for joining us on this webinar where we're going to be exploring some newish ideas and want to be thinking about the implications of real-world data and real-world evidence for clinical trial data sharing. This is a webinar hosted by Vivli. Just wanted to take just a short introduction to what Vivli is. It is both a nonprofit organization that really is it's a convener. It's a neutral broker among many stakeholders, concerned about sharing participant level data from clinical trials. So we convene people from groups from industry, nonprofit funders, government, and patient advocates. We do a lot of work around governance and policy. Not surprisingly, much of our value, I think, is from harmonizing language and agreements. We advocate for lowering barriers to data sharing, and of course, we are overseeing a data sharing platform where we are hosting data from trials on any disease from any country sponsor or funder. That's the context for what we're thinking going forward in terms of clinical trial data sharing, that Vivli has been designed for sort of the traditional clinical trial. And as we know, the landscape is changing very rapidly. Next slide, please. And so we wanted to take this opportunity in a webinar to explore with the community, with all of you and with our speakers, what real-world data and evidence is and how this fits in. So the objectives for this webinar, I will cover just the basics of real-world data and real-world evidence and review special types of real-world data. Marcia Levenstein will talk about real-world study designs and the type of questions they are suited for. And then we'll move on into an overview of the value of real-world evidence for regulatory science, and also a case study of using real-world evidence for FDA approval. And we'll have time at the end for questions as well. So Vivli, by the numbers today, there are over 4,900 trials available now on Vivli, representing over 2.7 million participants. We have 23 members from industry, academia, and so forth. Next. And looking forward, the real-world evidence, this is a definition from the FDA. Real-world evidence is clinical evidence about the usage and potential benefits or risk of a medical product derived from analysis of real-world data. So we have RWE, we have RWD, we have real-world evidence defined as clinical evidence. So I think it's helpful to tease apart a little bit more about what evidence is exactly. And for that, next slide, we need to have some better understanding of a distinction between data, information, and knowledge. So this is the triangle. Many of you may have seen this, that data are raw observations. They're objective facts that we observe in the world. Information is data within a meaningful context. And knowledge is understanding about the world. It's a statement about the world. It could be explicit, like a guideline. It could be tacit and not codifiable, so that it could be expertise. It could be how to do things, like how to put in a central line. And knowledge is really different from data and information, because knowledge helps you to explain, to predict, and to guide future action. Right? So this is all kind of abstract. So here's an example. So an observation of a hemoglobin A1C of 10.1, that's data. It's an observation. It's a raw observation. If you put it in context, that it happened last Thursday and 10.1% is far above the normal, that's information because it's data in context. And what does this tell you? It tells you that, because you have knowledge that high hemoglobin A1C occurs in a disease called diabetes, and it's associated with higher risk for cardiovascular outcomes, that then allows you to take this piece of data in that information context and allows you to make some statements about what might happen in the world, right, because of your knowledge. For those of you who are curious about what's at the top of this pyramid, next. It's wisdom, and that's another webinar that I'm not sure whether Vivli will host or not. But the question, of course, then is, well, where does evidence fit in? We've talked about data, information, knowledge. So next, where does evidence stand? Next. Evidence is the basis for a claim of knowledge. It is not knowledge itself, but it's evidence together that allows you to state some statement about the world. Next. And evidence is made up of three components. It's the data, it's the study design or how you collected that data, and then how you analyze that data to generate some statement that you can then use to claim, as support for knowledge. Next. Many of you are familiar with this sort of evidence equation where we have the best evidence, the sort of the top of the pyramid in our evidence-based medicine pyramid, where we're collecting data per protocol, right? So that, and we use the RCT design where we're controlling for all the confounders and the biases, and we do intention to treat, or IPD meta-analysis, if you, agree with that being the peak of the pyramid. You're all very familiar with this, I'm pretty sure. Next. But this evidence equation is changing dramatically, and we can just move through, Gabriella. Next, that we have, right, and one more, okay. That we have, the data is changing. We have big data, genomics, imaging. We have real world data, which I'll talk about in a little bit more. We have real world designs that we'll cover later in this webinar as well. And of course, lots of new methods of analysis that are, machine learning and others, data mining, predictive and analytics, just a tremendous amount of new analysis. So each of these equations are changing, which adds up really to a new world of evidence. Next. And so not only do we have the best sort of evidence-based medicine equation that we're so familiar with, but we're moving toward a world of fit-for-purpose evidence where we have real-world data, real-world designs, and data science. We can ask different questions. We can ask get different kinds of answers. And that's, I think, really forcing us to think about what kind of evidence we want, what are the claims we want to make, and how do we want to build the system where we can share the fruits of all those studies so that we can maximize our learning. Next. So I'm going to dive a bit more down into real world data. Next slide. So this is just a very, very quick overview, just to give you a flavor. When we talk about real-world clinical data, often talking about electronic health record data. This is data from real-time clinical care. Taking as an example, Epic, one of the most widely used electronic health record systems. Data is stored in Epic in an internal Chronicles database that is actually very hard to get at. So most of us who are using Epic data, it's a subset of data that is put into a queryable SQL database. And most times when we're querying, data from real-time clinical care, it would be something like the Clarity database, and there are, equivalent approaches for Cerner and other electronic health records. Often when we're trying to look at data to do research, it's not going to be the sort of real-time, runtime clinical data that we're looking at. We're looking at clinical data warehouse data, and that's data that is from the EHR, but it's sort of copied and sort of stored off in a different, in a warehouse, right? So that then you can go to the warehouse and ask different kinds of questions, like QI questions or secondary clinical research questions. An example here would be the OMOP. Common data model, so that's a standard database design for data warehouses that support QI and secondary clinical research. OMOP is the model behind the Odyssey group, which is a very large industry and non-industry group worldwide that everybody sort of agreed on a data model so that, once you put your data from an EHR into it, there are common tools and shared tools and ways that we can put data together. So that one is perhaps it's very powerful for reusing data, and you see a lot of real world data, real world evidence projects on the Odyssey platform. As data moves back and forth between EHRs and other health IT systems, some of you may have heard of FHIR, the Fast Health Interoperability Resources. It's a standard format for sending data from one system to another. So it's not a standard, it's not a model for how data sits in the database, but how it gets from one place to another. And of course, that's really important because we do have data or should have data flowing back and forth. So very quick review of sort of the types of clinical data when we talk about real world data. Next step. Do want to introduce also, claims data, of course, that's a huge one. And just these are examples, and we don't have any relations or conflicts with any of these systems that I'm mentioning as an example here. The Optum's data warehouse has 160 million lives across covering claims and clinical information, as you see here. Very, very powerful. So there are groups that have these sorts of claims databases that one could access and use for real world evidence projects. Next. We're rapidly moving into a world where electronic health data is going to be more democratized. Apple Health, if you have an iOS phone right now, you can get your data from over 600 health systems. You can just download your medical record onto your phone in about 20 seconds. I'm part of a project called Common Health that is building the equivalent for Android. And so there will be the day very soon in the next couple of months where with either Android or iOS, you will be able to get a good chunk of your electronic health record data, your labs, your allergies, your problem list, your encounters, and that's all going to be on your phone. It's not up in the cloud, it's on your phone, and it's going to be available to other apps on your phone. So that's really a sea change, I think, in the availability of real world data from clinical care. Next slide. Just wanted to cover also patient-reported outcomes. You're familiar with scales like PHQ-9 for depression and so forth. You're familiar with, these questions where there's a STEM question over the last 2 weeks, and then, do you have little interest or pleasure in doing things, and then a bunch of numbers that you choose from, right? Not at all, several days, and so forth. So very familiar with that. But I do wanted to say next that, we are moving beyond this. In digital health in particular, is something called ecological momentary assessments that refer to a collection of methods used really to ask you how you're feeling in the moment in your natural environment. Next. So an example of an EMA would be something like, where are you right now? Are you at home? Are you at a bar? Who did you drink with? This is obviously about alcohol. Did you drinking with a romantic partner or with a friend whom you haven't had sex with in the past 90 days, right? So this is really an immediate way of getting real world data that's really close to people's lives and people's experiences. This is something I think we haven't had before. It changes the kind of questions that we can ask, and I think it generates evidence that perhaps may be more relevant to our patients directly. Next. And we could even move on beyond questions that are in text and move to image-based ones. This is the photographic affect meter, where you just tap on the picture that you feel right now to reflect your mood. So lots of exciting things going on there. Next slide. And lots of exciting things going on in digital biomarkers as well. Sensors that can measure, obviously, your blood pressure, your heart rate. There are ones that combine ecological momentary assessments and sensors to see whether you're smoking, whether you're brushing your teeth, whether you're craving drugs, and of course, environmental sensors in the Internet of Things as well. So big changes coming on. Next slide. That's a very quick overview of sort of the real world data that's input into real world study designs. Next. That Marcia will talk about, and so I will hand over to Marcia at this point. Thank you.
Thank you, Ida. We can go to the next slide. I'm going to focus on really talking about the design aspects of real world data versus randomized clinical data. In terms of thinking about why you might want to use one kind of study design compared to another, we really want to think about what questions are we asking and what do we want to answer with clinical research. So we can do efficacy studies or we can do effectiveness studies. The efficacy studies are those that are typically performed using randomized clinical trials. The question they're answering is, does the intervention work under ideal circumstances? And that's important because they're ideal circumstances. They are very resource intensive in terms of getting this ideal setting. They're also very selective in terms of the patient population, resulting in a homogeneous patient population. The investigators tend to be highly experienced and trained, and the interventions the timing of dosage, the dosages that you use, the variables that are collected are all highly regimented and standardized so that we in fact can isolate the differences and make the best and strongest comparisons. In contrast, an effectiveness study asks the question, does the intervention work in real life practice? And as a consequence of trying to answer this question, these are performed in real life, everyday clinical settings. You wind up with a more heterogeneous population that reflects the clinical population that uses the drug or intervention. There are few exclusion criteria. The investigators are often much more heterogeneous and more spread out, not necessarily well experienced. And the interventions are applied with flexibility, and that really reflects both dosing, timing, and the variables that are collected. Next slide. So some of the characteristics of these studies will reflect on how we get the answers that we're looking for. So again, as I said, efficacy studies maximize your chances of seeing interventional effect if one exists. So efficacy is well-defined, your efficacy measurements are well-defined, your population, your timing and doses of medicine or intervention are well-defined, and the variables are well-defined. Everything is rigorously defined and needs to be met for this to be a randomized controlled study that will really isolate the differences. Next slide, please. Real-world trials, in contrast, take into account external factors such as patient factors, provider factors, and system factors. Those, in fact, may make it harder to distinguish the effect of the intervention that you're studying. And again, this really is through a real-world, very clinically based setting of both the intervention, how it's used, when it's given, the variables that are measured. They need to be clinically relevant. Those are ones that would typically be collected in a clinical setting so that the clinicians can use them and are accustomed to them. They would be measured to really account for how the patient is doing in the real world. Therefore, these may vary over the patients, and it makes it, again, more difficult to distinguish differences between interventions. Next slide, please. Now, when we talk about real-world studies and randomized clinical trials, it sounds like these are two very distinct kinds of approaches to answering clinical research questions. In reality, there's a continuum. You start at the most rigorously and highly defined randomized clinical trials, and you wind up at the end with something that's really purely observational, has very little predefined in terms of interventions, timing, and variables. So you get from the most controlled to the least controlled. And we'll go through some of these. I'll show you a little bit of this hierarchy. And then when we've gone through these kinds of studies, I'll focus a little on some of the key studies that we'll talk about and you may think about. So we start with randomized clinical trials. And again, then we have meta-analyses, systematic reviews, decision analyses. There are both prospective and retrospective cohort studies, case control studies. Next slide, please. Cross-sectional studies, ecologic studies, pragmatic studies, and or large observational studies, case reports and series, and then registries are really very observational in nature and have very few controls, as you would see in terms of a randomized clinical trial. So next slide, please. So to give a little more detail about randomized clinical trials, these can actually show causal relationships and establish superiority of efficacy among treatments. So with many studies, you may be able to see relationships and correlations, but you can't necessarily determine whether or not there are causal relations. Because in a randomized clinical trial, you've standardized so much of the setting and the interventions, what's left is really attributable to the intervention under study. They're very resource intensive, and the generalizability may be limited to the study population because you've set in place so many constraints on who may enter the study, what they can do, what other drugs they can do, and how they're treated. But clinical trials have defined cardiovascular benefits of lipid-lowering drugs, blood pressure-lowering drugs. They've also been used to establish efficacy for pretty much all the drugs or interventions that are currently being used in patients around the world. Next slide, please. Now, a pragmatic clinical trial or large clinical observational studies can generally be well extended to large populations, heterogeneous populations, again, that are typical of the patient population that uses the intervention. They are relatively low cost. And again, I say relatively because all studies, if they're large and collect information, are not inexpensive, but these are low cost relative to controlled clinical trials. And again, they really have much increased external validity because you've put in place many fewer constraints and limitations on the participants of the study. As a consequence of all this flexibility and the ability to reflect clinical practice, data quality may suffer. Lack of standardized treatments are in place. Assessments and adherence may also be much more variable, and therefore this weakens the ability to be able to statistically see a treatment difference if one exists. It needs to be large enough to overcome the drop-ins, the dropouts, the crossovers, the differences in timing of when variables are measured, perhaps the actual variables that are measured, differences in how practitioners will measure these variables. But these, again, can be very strong, and they have been used to show clear evidence for clinical decision-making in type 2 diabetes, especially with respect to the use of sulfonylureas. Next slide. And so the last study type I'm going to talk about is registries. And again, registries are determine efficacy in clinical setting. They can be very useful, particularly for rare diseases. If you're studying a rare disease population, it may be difficult to find these patients. So being able to focus in on registries of clinicians who are treating these patients in large numbers can be very helpful in collecting large amounts of information, or relatively large amounts of information, efficiently and effectively. They can also be used to look at quality of care, and you can get results relatively rapidly because you're able to focus your studies in settings where there are relatively large numbers of patients. But confounding and bias may not be controllable, in large part because you don't understand why a patient may or may not receive a particular drug. Because they're in clinical settings, decisions about which interventions are used and what in fact is specifically done for each patient, how dosages are increased or decreased, depend upon clinical practice and the individual decision-making of the physician. So you may not be really understand what's behind the decision-making. Consequently, you can't distinguish and adjust for biases. But these have been used to determine predictors of survival in rare diseases such as pulmonary arterial hypertension. And again, this allows for making better treatment decisions by clinicians in the real world, which is critically important for patients. Next slide. So all the studies we've talked about thus far really are studies that require primary data collection from patients in real time. You can also look at electronic health records or insurance forms as another way to do secondary data analysis. Use existing data to answer clinical questions. This again is a much more efficient process from a time perspective because it's clearly more efficient to use data that already exists rather than having to go out and collect clinical information. Some of the examples really echo the kinds of data that Ida already described. You can use claims forms from insurance companies. You may also be able to use hospital, clinical, and usage data. Again, not necessarily in real time, but repositories of this kind of information. Often there are national or governmental databases, such as those in Scandinavia and the UK, that have information that can be very useful for registries and other kinds of studies. You can also, if you have sufficient information and similar sources, you can merge clinical and economic data from different sources to answer the questions that you have, particularly about effectiveness and relative effectiveness. Next slide, please. There also are hybrid study designs. These are studies that are not necessarily either completely randomized or completely real world. So you can have randomization requirements and perhaps patient inclusion exclusion requirements, but the use of pragmatic outcomes are very little oversight, much flexibility in terms of how the studies are performed, what dosages patients receive, how often they come to the treatment, to the centers, and what the observations are. So it gives you elements that are both controlled and pragmatic. You can also have single-arm treatments in randomized in clinical studies that are, again, well-defined with well-defined populations and clear restrictions and requirements on the timing and dosage of visits. But you may supplement it with external comparators from real-world data or existing clinical trials. You can also use real-world data in a randomized clinical trial. As Ida has described. And again, also from the analytical perspective, you can combine real world data with randomized clinical trial data to, again, get the best of both worlds. So next slide. So this really leads us to our last question, which is, now that we understand better some of the differences and what kinds of questions you can answer and ask with these kinds of studies, is it feasible to use real world data to replicate what can be done in a clinical trial? So a study was performed to really identify the numbers of clinical trials that were published in high impact journals that could be performed instead feasibly by using existing data insurance and EHR systems. Go to the next slide, please. You can look through this flow chart. You can see that they started with 220 studies that met the criteria. And as you go through each of the requirements to see whether or not the information exists in the secondary sources, you start with 86 have an intervention that you could identify within an insurance or electronic health record source. 62 of those had indications that you could define. 45 of those had a large percent, that is 80% of the inclusion-exclusion criteria could be determined. And ultimately, there were 33 studies of the 220, that's 15%, that really you could have replicated using real-world data rather than having conducted a randomized clinical trial. The next slide. So then the question becomes, where is this really most useful? How can we take advantage of the strengths of real-world data compared to randomized clinical trials? If you look at the kinds of interventions that were studied or that they found in these studies, you can see that in the second line, that is for those interventions that were seeking FDA approval, they were not yet approved, you couldn't replicate any of them. And that really echoes one of the strong differences between real-world data and randomized clinical trials. In order to perform real-world data, people need to be using the intervention in the real-world clinical setting. So that's an important thing to understand. Conversely, in the post-approval safety or efficacy setting or in comparative effectiveness, nearly half of the studies could be replicated using EHRs or insurance databases. So I think this gives you a good idea of how you can position these different kinds of studies and different kinds of data to answer questions and make important decisions in the world of interventions and understanding clinical research. So now I'll hand it over to Greg, who's going to talk about the FDA perspective on these two kinds of studies.
Thanks, Marcia. Let me see, am I on? Yes. I want to thank Ida and Vivli for the invitation. I'm a little self-conscious. I'm not going to tell you what FDA's perspective is. There's a bigger subject. Next slide. This is really a case study and some experience and that's evolving at the FDA. Next slide. And I want to change the lens of the discussion a little bit to talk more about obviously the regulatory context. I found an article by Matt Herder in Milbank Quarterly very useful in his discussion of 21st century cures, making the point that the regulatory landscape has changed dramatically over the last several years with orphaned designation, accelerated pathways, breakthrough designations, making the pre-market efficacy decisions based on comparatively few case studies. There are major decisions made on new products that are being made on 6 or 7 patients, and that can be very appropriate given the context, given the question being asked, efficacy. Where does that leave us regarding safety? And that's really the shift of the lens here. 21st century cures gives us very clear direction, authority, and money to look at real-world evidence, but it focuses it really. And here I haven't read some language from the law. It focuses our efforts on expansion of labels, new indications, and supporting and satisfying post-approval study requirements. So it really is very much on the post-approval side, and on the as I said, safety becomes a major consideration. Next slide. I'm not going to say anything about our limitations of current safety programs at the FDA. Those are well understood, and the limitations are well understood. I see 21st century cures and real evidence and new things evolving as augmenting systems that have known weaknesses and helping us move forward. What we are, and my colleague, I want to first of all shout out and thanks to my colleague Danica Marinik-Davic, who a lot of this work that I'm going to be discussing today is her effort, and with her colleagues at MD-EpiNet, Medical Device Epidemiological Network, a public-private partnership. And they always advise me, don't talk about pre and post, talk about total product life cycle, and that's quite correct. There is a change of the balance between pre and post within that total product life cycle. Next slide. Dr. Abernathy, our principal deputy, who's provided us with such great leadership in this area, talks about it as the snail. This is her snail. Next slide. And what we have is a shift where the pre-market area, and this is the blue steps, becomes smaller, and we rely as approvals move forward in time and with less effort, less sample size, less study size. Next slide. Next slide. And we have you see this shift that's taking place, and that's the head of the snail is getting smaller and the back of the snail is getting larger. Next slide. Now, to demonstrate this in a case study, actually it's a couple of case studies, one published, one unpublished. It's going through the process now. I'm going to first rely on a paper that appeared last summer in the BMJ, looking at TVT, transcatheter valve therapy, and the use of coordinated registry networks to evaluate that device and that procedure. Just a little tune-up or a little slight difference of perspective or use of words from Marcia. I'm using the word here registry not as a methodology but as a data source, and a coordinated registry specifically is a registry, traditional registry, those primarily that are being collected by national specialty societies that are then augmented or linked through linkage with other real-world evidence, or as Marcia very rightly pointed out, through hybridization by linking it with data that's collected specifically for a particular study. Now, the magic on this, from my perspective, is that the data in the registries that we're talking about are collected for many purposes. There's many stakeholders. Next slide. And the mantra for value is collect once, use many times. Data is expensive. There's no getting around it. There are ways I'm going to talk about that will drive down the cost in the future. But when you collect data once and use it many times, this is where the value currently is being generated. So the TVT case study originated in the American College of Cardiology plus the Society of Thoracic Surgeons registry, now 20 years in existence, that originally came up as a quality improvement effort. It evolved to support clinical research and development of products, and ultimately, in this case study, it documented the use of that data set linked to CMS claims records for FDA purposes. The same data set actually was used for a national coverage decision. Next slide. And jumping ahead to the findings, I'm not going to spend too much time on the study. You have the citation if you'd like to look at it. The return on investment of this effort was over 500%. The salmon-colored bar is the cost that 3 companies paid to the registry to conduct studies for them. The registry, the National Specialty Society functioned like a CRO in this context, and they received about $25 million to do a series of studies, about 29 studies. And the blue bar represents what we're calling the counterfactual, the cost of what it would have taken if we had done traditional studies. And these traditional studies are post-approval studies, mandated post-approval studies that were required as a condition of approval, and then label expansions. There are also studies done for compliance, FDA studies. So this was a very useful, robust data system. And as I said, it connected the, so what's an epidemiological study? It's a cohort that was collected by the registry of almost all comers. This is like 97% of all cath labs contribute to this. So the last I checked is well over 200,000 patients had been enrolled over time in this database. And then the intervention, and that's the specifics of the TVT, that's obviously collected in the registry. Typically, registries in national clinical societies have only a 30-day follow-up because they're for perioperative problems and those kind of quality assurance issues that hospitals are concerned about. What the FDA was encouraged industry and ACC to work together to then link that data to CMS claims data to give them longitudinal follow-up data that accrued over time. We linked the data, and I'm not going to say anything about the validation of endpoints, validation of data sets, validation of methodology. That's all in the literature that you can find at the MD EpiNet website. But anyway, you see this substantial cost savings, return on investment due to this mantra that I've used of collect once, use many times. And I'll say something bold, this is my personal opinion. For real-world evidence, if we're not making it better, faster, cheaper, I'm not sure why we're doing it. We have methods to do these things otherwise. Next slide. Just a quick methodological note. To calculate the cost of the counterfactuals, we use a study that was done by Fred Resnick, a model, and those of you who do clinical trials, or do these kind of studies, will recognize these as what fuels or generates the cost of the studies: the recruitment versus randomization, controls, the number of sites out of the United States, then did you call the person, how many procedures, what kind of organ system, all these things are the major drivers, and this is what goes into the this is the model that produced the counterfactual cost of what these studies would have cost if we had not used the CRN, the Coordinated Registry Network. Next slide. Very briefly, we've got one other manuscript under review now, looking at vascular devices. Here we worked with the Vascular Quality Initiative, a quality assurance process that was spurred by AHRQ, that brought together the various professional groups that conduct or that provide services of vascular surgery services, and we used that cohort and then linked it to a data that was collected specifically for this study. This is a group of the magic, of course, of the TVT is that we're using CMS data, which gives you information on everyone who has who's over 65. For vascular surgery, and that's for the TVT, that suffice. For vascular surgery, there are many patients that are under 65, so we don't have that resource. This was actually in the evolution of this coordinated registry network evolution. This was an earlier study, and this was really a traditional post-approval study, actually a series of them, about 7, that were nested within the registry and then traditional follow-up mechanisms. But even with that methodology, 4 companies were able to save substantial amount of money and a return on investment of over 140%. Next slide. Now, I have used the stream better, faster, cheaper to describe the desideratum of real-world evidence, and the time in the paper. You can look at the specific methodology, but I'm going to summarize it here. It's very easy to understand time savings. The typical post-approval study or clinical trial will have 30, 40 sites at the max, and accrual of patients in that setting is very slow. It can take years, depending on the particulars of the study. In the ACC setting, with 900 sites reporting, lickety split, you get enrollment, and from months to years of time were saved on these studies that were done on the TVT. Now, again, with the vision study, with the vascular studies, the time savings was measurable, was very real, but it was due to the nature of the registry. You can think of it as a platform trial, a series of sites that were ready to go without a lot of problems, issues regarding enrollment or IRB, startups, and the follow-up was done in the traditional way. So we didn't have time saving there, and the number of sites was as with a traditional post-approval study. And again, as they say, this earlier exercise in the evolution of this methodology was a traditional post-approval design nested in this activity. Next slide. Just briefly, the public health benefit of this. The better, faster, obviously, industry finds that great, and public health should too. We like to get we like to be efficient, and we like to get the products and the decisions made faster. From a purely epidemiological effect, having these large sample sizes, study sizes, as with the TVT, provides us with a better understanding of heterogeneity. We have a lot of patients here, a lot of differences that we can look. Just as a note here, propensity score matching using the controls of people who did not receive the device are very, very useful in this setting. Next slide. Futuristically, where can this be going? How can we see this evolving? Let's look at two different buckets: the follow-up bucket and the platform bucket. There are lots of things going on in the real-world evidence or health data, I guess, arena broadly. There was a Senate committee. It made its way to a draft bill that was part of the medical surprise bill. It didn't pass, but we'll see what happens next round. But an all claims database for the nation is being considered. Actually, the purpose of it would be to study drug costs, but the same data could be used as follow-up data for a host of real-world evidence efforts. Already 17 states have all claims databases, and building on that would provide a great resource for efforts like the CRNs that I've been mentioning. Also, as Marcia beautifully put out, there's all sorts of exciting, Marcia and Ida, efforts going on around PROs and EHR, making them more accessible, and mobile apps to provide information on follow-up. On the platform bucket, ways to lower costs are already being explored, and some of the registries are moving away from traditional curation by an army of nurses with clipboards and rule books towards a ingestion of data from a hospital, and then centralized curation supported by AI and other advanced technology to improve the quality and decrease the cost of that effort. I won't say anything about natural language processing, but that obviously is another tool that's being considered. Next slide. The coordinated registry network is a data source that is evolving. Currently, there are about 15 under development in the device space, which covers the majority of high-risk devices that are regulated by the FDA. You can think of Flatiron as like a commercial version of a CRM. I mean, it's a very similar kind of effort that's used quite extensively to evaluate cancer drugs. And very recently, I've had the pleasure of working with the American Society of Hematology to establish a coordinated registry network. They're calling it a data hub because it provides both a clinical trials network for for pre-approval, for approvals, initial approvals, pre-market, and also the post-market setting data, observational data, traditional coordinated registry approach. And they have, they're standing up these efforts for both sickle cell disease and multiple myeloma, both around which there are very exciting new cures on the horizon. Next slide. So with that, I want to thank you, and what a pleasure and honor to be asked to speak with Marcia and Ida today.
Great. Thank you, Greg. Now we're going to turn over to Jack Markadian to talk through his case study. Over to you, Jack. Thank you.
Okay, great. Thank you. And welcome, everyone. I'm going to present a case study on the use of real-world data and the resulting real-world evidence that was summarized in a successful regulatory submission from Pfizer to expand a palbociclib indication to males with metastatic breast cancer. I was a member of the submission team, and for your information, the brand name of palbociclib is Ibret. Next, please. You heard from the previous speakers describing the various sources of real-world data and the promise of real-world evidence despite its limitations. What I'm going to do is basically describe the submission and the external feedback based on postings from the internet. I'll summarize the FDA feedback based on an FDA approval summary that was published as a manuscript, and I will reference that for you, and I encourage you to read that manuscript. I'll end with my own thoughts and leave you with hopefully some time to ask questions. So breast cancer in men is a rare disease, especially in the United States. Less than 1% of all breast cancers occur in men. In 2020, about 2,600 men are expected to be diagnosed with the disease. Due to the rarity of male patients with metastatic breast cancer, it's not feasible to conduct large, adequately powered randomized clinical trials. The objective of the regulatory submission was to incorporate real-world evidence, including use, clinical activity, and safety data of palbociclib to assess the benefits and risk of palbociclib plus endocrine therapy in men with HR-positive, HER2-negative metastatic breast cancer. Basically, we were pursuing a label extension that was based on real-world evidence. We had several meetings and interactions with the FDA where there was demonstrated enthusiasm and interest from both parties for the submission. Especially useful was the involvement of the Flatiron team in preparation for the meetings. Next. One real-world data source that we used was the IQVIA claims data. And essentially, the IQVIA pharmacy and medical claims data source was analyzed to assess real-world usage patterns of palbociclib plus endocrine therapy by line of therapy, and duration of palbociclib was compared to duration of therapy of endocrine therapy alone. The medical claims were used by IQVIA to identify patients with metastatic breast cancer. So basically, we were provided pharmacy claims data from about 50 male patients who started palbociclib plus endocrine therapy in a first or second line setting, and approximately 240 non-palbociclib treated male patients who started endocrine therapy in a first or second line setting. The data provided were basically medication start and stop dates based on prescription fill information from the IQVIA database. Age was the only patient information provided. We relied on duration of therapy as a marker for progression-free survival. We analyzed the data descriptively. The median duration of treatment was estimated using Kaplan-Meier method. The Kaplan-Meier curves that we showed in the submission basically demonstrated that median duration of treatment in the first-line setting was longer in the palbociclib cohort than in the non-palbociclib endocrine therapy cohort. We didn't have baseline characteristics, so we decided not to conduct typical comparative effectiveness analyses that rely on methods such as mentioned before by Greg, propensity score matching, to balance baseline patient characteristics. Next. The second real-world data source was the Flatiron electronic health records. As expected, the electronic health record data that we received from Flatiron was much more rich in its depth. De-identified outcomes that were extracted by Flatiron included tumor response based on clinician assessment, duration of treatment, and targeted adverse event data. However, only 59 patients, 29 palbociclib and 34 endocrine therapy patients were identified. Among the identified patients, a further attrition was due to the fact that only 28 patients had tumor response assessment during the study, and this is reflective of real-world practice compared to clinical trials where protocols require scheduled tumor response assessments. The data were analyzed descriptively. Median duration of treatment was estimated using the Kaplan-Meier method. We used swimmers plots. Patient narratives included both text along with patient journeys that were displayed graphically using a system that was developed by Flatiron. Next. So we received good news on April 4th, 2019. The FDA expanded the approved indications in women with HR-positive, HER2-negative, advanced metastatic breast cancer in combination with AIs or fulvestrant to include men. The FDA approval summary indicated that basically the approval was based primarily on the results of the PALOMA-2 and PALOMA-3 clinical trials that were used in the submission for females, and the keyword supported by real-world data from electronic health records and insurance claims. Again, I recommend reading of this clearly written manuscript and well-written manuscript. Next. The real-world use by FDA received positive press, and what I did here was just give you one example of a posting from the GNS blog, and there's just a few highlights I want to share with you. Again, the keyword moving the needle with real-world data, how oncology is leading the way. Real-world data has led to several key development successes for pharma, including label expansion. The approval was based almost entirely on analysis of real-world data from EHRs and post-market reports. It is true that real-world data motivated the submission. A big win for the IBRANCE team, particularly when it comes to expanding treatments for rare diseases or presentations, and this is with respect to the use of real-world data. The case offered proof that the use of real-world data can help accelerate approvals while maintaining safety and efficacy by reducing the need for time-consuming, expensive clinical trials. Next, please. The feedback from FDA was a little bit more conservative than what was posted on the internet. The Flatiron analysis initially identified 2,675 male patients. After additional data processing steps, we had 28 patients available for analysis. Again, this is typical of real-world data because from that 2,675 patients, we had to basically select patients with activity in the system, patients with structured orders for palbociclib or endocrine therapy, They needed to be confirmed HR positive, HER2 negative, and then we used unstructured data basically to confirm that they actually received the treatment that they were assigned. So in the end, we only had 59 patients, and among those, only 28 had tumor assessments. For those of you who do these kinds of studies, I think you realize that this type of attrition is not uncommon in real-world studies, but it shows you some of the sensitivities of the FDA. They think in terms of bias with such attrition. The baseline characteristics were not well balanced across the two cohorts, but this was not to be this was not very surprising. Recall the data on males was recorded prior to approval. Next slide, please. Basically, I'm sorry, can you go back one? I just want to, from the pink sheet, from the pink sheet, the key point from FDA's perspective is that conclusion of substantial evidence of efficacy was in fact based on Pfizer's 2 pivotal breast cancer studies, which were submitted for approval with the original female-only indication. And again, it's very clear, we still needed substantial evidence of effectiveness, but again, it was stressed that this was very interesting for us to review this application. She also suggested important experience was gained from inspecting the sites in the real-world studies. Again, this emphasis on assuring an appropriate audit trail to ensure that positive observational studies are not simply cherry-picked. And again, you see some concerns from FDA. Next slide, please. My own thoughts, basically, the comments and questions on the review of the submission suggest FDA reviewers are looking for data analysis that is structured more similar to a traditional clinical trial. It is clear to me they're looking for evidence to correlate real-world endpoints to a traditional clinical trial endpoints. They would like us to more closely mimic RECIST criteria for breast cancer studies. We need to, however, continue efforts to increase the understanding of the capabilities and the limitations of FDA, limitations of real-world evidence for FDA purposes. We basically need to publicly post study protocols and study results. We need to document patient counts that are obtained prior to finalizing the statistical analysis plan, especially since we receive the real-world data from vendors. It's hard to get a handle on what we actually have in our data. So, for those of you involved in generation of real-world evidence, you know that you do not completely understand all facets of the data, and sometimes you write the analysis plan after you've looked at the data. This aspect may not decrease, may actually decrease the comfort level of the FDA. So I thank you for your attention. To end on an encouraging note, the FDA has made it clear they continue to discuss the potential for the use of real-world evidence as primary to support regulatory decision making. Thank you. I now turn it over to Julie, who will hopefully give us some time for questions and answers.
Great. Thank you so much, Jack. Really appreciate that. We are right at the hour. If the panelists could stay on for a little bit longer, we will turn over to some questions. If you do have questions, please could you put them in the questions box in the control panel. To just kick us off, I want to turn to the panel to just see if you have questions for each other. If we don't get to all the questions, we will, of course, try to answer you offline. So if you could put in your questions, but I want to turn to the panel now and ask the panel if they have any questions that they wanted to ask each other.
This is Ida. I'll kick off with one. I'm very enamored of the possibilities of patient-reported outcomes being a primary care doc. I think it's patients are very concerned about harms and about side effects, and whether these real-world ways of getting a patient-reported outcomes might allow us to address the question of harms and side effects, not just post-approval, but more robustly in the pre-approval phase as well.
Ida, I'll say something. In the coordinated registry network space, orthopedics has taken the lead. In the evaluation of orthopedic devices and medications and procedures, pain and satisfaction are always critical. And those have been added using mobile apps and other sort of advanced technologies to that evolving registry. So you're spot on in your interest. I think more good news in the future.
Thanks.
Great. Thank you. Now we have a question from the audience asking, can you comment about selection bias in registry and administrative databases?
Yes, so this is Jack. Basically, how to answer that is primarily, hopefully, through the methods, the analytical methods, right? We know it exists, and so hopefully we do something to correct that concern in the analysis.
Adding to the traditionally understood propensity score matching, there are people, maybe Ida can speak on this, that are using machine learning as a tool instead of a kind of similar sort of approach, but using artificial intelligence to address those issues. The limitations are well understood. If you have cohorts that don't really overlap, it's difficult to see them as contributing anything in a modeling setting. But when you have large numbers of descriptors and large numbers of populations, the possibilities improve, obviously.
Great, thank you. I want to move to another question. I think we may have to, this may be our last one, just given time, but we will come back to the other questions and get responses back to you offline. The question is, do you see clinical trial protocols changing to include real-world evidence arms?
So this is Marsha. I think it's already changing. So I do believe that with the advance of repositories of data and large sets of data, that companies are starting to look at opportunities to use placebo groups, perhaps from other existing studies, as an alternative to conducting a very large randomized clinical trial. So I do think that the opportunity, the existence of large databases of placebo studies, or perhaps alternative studies that you can look at the data and understand what the impact was, can be used effectively for as a comparator arm. And I think that people are also thinking more about electronic devices, certainly, the Internet of Things, collecting data electronically, including more clinically relevant outcomes even earlier in studies, is something I think that companies are looking at. And also PROs is an area in which companies are incorporating elements of real-world data into perhaps more traditional real-world randomized clinical trials earlier in the process, so they have information that will be relevant that people will be able to look at the time interventions are really looked at for approvals.
Great, thank you, Marsha. So I think, just in the interest of time, because we have run over a little bit longer, we will come back to anyone who has asked questions that didn't get asked, didn't get answered here. If you have other questions that you've thought of, please do add them in the questions box and we'll come back to them. Again, just want to say thank you so much to our panel for all of your time and effort and all the knowledge that you've shared around this important topic. We will circulate the slides and the recording shortly. So again, thank you everyone for your time. Much appreciated. Goodbye.